So first of all, I would like to thank to all of you who donated and who strived for this grant, and to say that it means a lot for me to be awarded with this grant and to contribute with my research. So my project consists in the evaluation of new anti-prion drugs, which were planned and synthesized for the purpose of this study, and to combine different approaches to find new drugs to treat prion disease. So, as you all have heard, the main event in prion disease is the conformational change of the prion protein, the normal prion protein, which we call PRPC or PRPSAM, to the pathogenic isoform, which we call PRPSC or PRPRAS. So the cells in our body produce the normal prion protein, but once the PRPSC is present, this pathogenic isoform is then able to convert the normal prion protein and make new copies of the pathogenic isoform, leading to the accumulation of PRPSC both inside and outside the cells. And the ones that are outside are then available to infect other cells. And this is how the disease is spread through the brain. A really important characteristic of prion disease is the existence of different prion strains, which means that the PRPRAS can adopt different conformations, which means different shapes. So, and each shape is associated with a prion strain. So in the lab, this means that we can find different results because the different strains have different behaviors. And biologically speaking, it means that these different strains will tend to accumulate in different brain areas, and this will lead to different clinical signs. So this is why it's so difficult to find a drug that works for different prion strains. So there are many reports in literature from different researchers showing that we can find a drug that works for one prion strain, but unfortunately it doesn't work for another. So this is why it's important to assess different prion strains. The drug discovery process for prion disease comprise different stages. So overall, more than thousands of compounds have been tested by many researchers in the world. And then, based on the initial results, the researchers propose some chemical modifications in the structure of the drug, aiming to find drugs with better characteristics, such as more with solubility. So then hundreds of them have been tested in vivo in prion-infected mice, but here is where they fail, and for many different reasons. And the main ones are because they are toxic or because they don't reach the brain, which is the target of this disease. And then to date, less than 5 drugs got to the point to be tested in humans infected with CJD. And unfortunately, none of them really worked. And this is why these diseases are still untreatable. So this highlights the difficulty to translate the results we get in vitro to in vivo to animals, and then from in vivo to humans. So, and another important thing here is if we are able to use human prion strains in early stages of research, this can increase our chances to find a drug that will work in the end in humans. So where did my project start? So a few years ago, I analyzed a library of 200 compounds, and by using different approaches, I found this lead compound that we call J8. And based on the structure of these compounds, my Brazilian collaborators planned and synthesized a new series of compounds that we call LCs. So the goal of my project is to combine different approaches such as prion-infected cells, cell-free assays, where I can test human prion strains, and the newly developed model of human prion infection, which are the cerebral organoids, and lastly, to test the prion-infected mice in order to find an anti-prion drug that will work in vivo, in humans, sorry. So the first model I used are the prion-infected cells. So these cells are derived from a mice tumor, and this is why they are able to grow indefinitely in a dish, and they are infected with prions. So this is what happens in the absence of treatment. We have a really strong signal of PRP-RAS, but in the presence of the different drugs, we see an inhibition of the accumulation of PRP-RAS, which is showing here in this graph. So here are the untreated cells, which have a lot of PRP-RAS, but in the presence of these drugs, they have few amounts of PRP-RAS. So by using cells infected with the RML strains, these drugs were able to reduce by more than 70% the amount of PRP-RAS. However, when I tested the same cell but infected with another prion strain, which is the 2N2L strain, only few compounds were effective, and even though their effect was weaker than in the RML strain, reinforcing that some compounds can work for a given strain, but they are ineffective against others. So then I switched to another model, which is a cell-free assay, where I could test a human prion strain. So in a tube, I added a brain homogenate from a patient with CJD, and I added the normal prion protein because, as I told you before, the infectious prions in the CJD brain homogenate are able to convert the normal prion protein, and this is what is showing here. I have a really strong signal of PRP-RAS, and here are the same reactions, but in the presence of the different drugs. And here, LC19 and mainly LC87 were able to reduce or inhibit the PRP-RAS formation in vitro. And again, here I used a human CJD prion strain. But we have to keep in mind that this is a cell-free assay. And in a cellular environment, many other factors can interfere with the interaction of the PRPSC and PRPC. So this is why it's so important to have a human model where we can test the efficacy of these drugs in earlier stages of the research. So here, I used the cerebral organoids. So I've been doing these experiments in collaboration with Dr. Catherine Hai and Bradley Groveman, who developed this new system. So these cerebral organoids, which are also called mini brains, are derived from skin cells collected from patients. So by adding different factors in vitro, we are able to induce the formation of this three-dimensional tissue, which have some characteristics of the human brain, and this is why we call them mini brains. So then, by adding a brain homogenate infected containing CJD prions, they showed that in the first month, they had low or no detectable levels of PRP-RAS, showing that these mini brains get rid of the inoculum. There was no residual inoculum there. But weeks later, they found new generated CJD, new generated, I'm sorry, PRP-RAS. So this is the most recent model of human prion infection. So then I decided to assess two different regimens. The prophylactic treatment, where we can treat these mini brains prior to the infection, which mimics what could happen in the context of genetic prion disease, where we can treat these patients before the appearance of the symptoms. So here, I started the treatment. I exposed these mini brains a week before infection to two different drugs, the J8, which is my lead compound, and PPS, the pentosan polysulfate, which is a non-anti-prion drug, which works in cells, in animals, but unfortunately didn't work well in humans. So, and then I removed the treatment. I exposure a week before the infection, and I kept for 2 more weeks, and then I stopped the treatment. And then a month later, I measured the amount of PRP-RAS in these mini brains. So in the first month, I couldn't see any big difference in the treated and the untreated mini brains. But 2 months later, I saw that the mini brains, which were treated with PPS, they had tenfold less PRP-RAS compared to the untreated ones. The J8 inhibit a little bit, but it's not significant, statistically significant. So then I assessed the late treatment, which is the therapeutic treatment, which is similar to what happens with CJD patients, where we treat these patients, but the disease is well established. So they have the clinical signs of the disease. So here I start the treatment 2 months after these mini brains were infected. And then I treated these mini brains for a month, and I measured the amount of PRP-RAS. And as we can see here, both PPS and J8 were able to reduce the amount of PRP-RAS in these mini brains by more than 100-fold. And then 120 days post-infection, which means 2 months later, even though these mini brains were not being exposed to the treatment, the ones that were treated with PPS still had more than 100-fold less PRP-RAS than the untreated ones. And the ones which received the J8 also had less prions, but again, this was not statistically significant. But we have to remind that this was just a brief treatment. Now we are performing this in a long treatment all the way to the end. So this highlights the utility of this new model of human prion disease to access the efficacy of anti-prion drugs. So to summarize my data, we showed that these three-dimensional cell culture, which we call mini brains, are useful models to verify the ability of anti-prion drugs in inhibit the conversion in a human background. And we hope that by using this human model, we can increase our chances to find anti-prion drugs that will work in humans. So our future goals are to test the LC's compounds in this system in the mini brains infected with CJD, and lastly, to test the effective drugs in humanized transgenic mice that are able to be infected and propagate prion infection when they are inoculated with CJD prions, which are human prions. So I would like to thank you again to the CJD Foundation, to the families who contributed for this grant. So the Dodom Dorf, the Jeffrey Smith family, as well as the people who strided for this grant. Thank you all. As well as my Brazilian collaborators who designed and synthesized these drugs. So Irama Cordeiro, Maria Letícia Barbosa, Arthur Camerli, as well as Bradley Groveman and Catherine High, who taught me how to grow and generate these mini brains in the lab. And of course, Dr. Colhay, Byron Colhay group, where I work at NIAID. Thank you all.